Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the receptor tyrosine kinases. Okay, the receptor tyrosine kinases. And first of all, we have to know that this receptor tyrosine kinases is coming from what? Okay. First, in my previous video, we have learned about the enzyme linked receptor. That means three kinds of receptors are present within the enzyme linked receptors. One is the receptor tyrosine kinases, another is the tyrosine kinase associated receptor, and the third one is the receptor serine or threonine kinases. Okay. So here the first is that receptor tyrosine kinases. Okay, and this is the one type of receptor under enzyme linked receptor. Okay, and before going to this topic, I want to say thank you to all of you who have already subscribed my channel and support me a lot. And I also request to the newcomers, if this channel is helpful to you, obviously share and subscribe this channel for giving your support to me. Okay, so let's continue. What is that receptor tyrosine kinases? That means this receptor tyrosine kinases are the second major type of cell surface receptor. Okay, so this is the second. Why? Because first one is the GPCR, and I have already uploaded about GPCR receptors in my channel, and so you just check out that video. The ligand for RTKs, the short form of receptor tyrosine kinases, that is the RTKs. Okay, the ligand for RTKs may be, so here in case of receptor tyrosine kinases, there are different ligands are there who can bind easily with the RTKs, and these are like insulin, epidermal growth factor, transforming growth factor alpha, platelet derived growth factor, fibroblast growth factor, and colony stimulating factor 1. So these are the ligands that have the quality to bind with these RTKs. Clear? All the RTKs generally comprise of an extracellular domain containing a ligand binding site. Okay, ligand binding site. So what is there? All RTKs, they have an extracellular domain containing a ligand binding site and a single hydrophobic transmembrane alpha helix and a cytosolic domain that includes a region with protein tyrosine activity. So what are there within the RTKs? First of all, one domain, obviously extracellular domain, that containing ligand binding site, one single hydrophobic transmembrane alpha helix, that means this is the alpha helix. Okay, alpha helix, and a cytosolic domain that containing a region for what? For the protein tyrosine activity. Okay, so these are the parts that remain within these RTKs. Most RTKs that are monomeric, okay, they remain singly, they remain in singly binding ligand, they remain as ligand binding receptor singly in the plasma membrane. Okay, all the ligand binding to the extracellular domain that induces the formation of the receptor dimers. So what happening when the ligands, suppose these are the ligands, when these ligands bind to this ligand binding site, so what's happening? They remain in first condition, or before the binding of ligands to the ligand binding site, they remain singly or they remain as monomer. Okay, but when the ligands bind this site, they remain as dimer. Okay, or you can say the receptor dimer is formed due to the binding of the ligand at the ligand binding site. The ligand binding leads to the activation of the intrinsic protein tyrosine kinase activity of the receptor and autophosphorylation. So what happening when ligand bind to these receptors? What is happening? The activation of the intrinsic protein tyrosine kinase activity, okay, of the receptor and autophosphorylation of tyrosine residues in its cytosolic domain. Clear? So two things happen here. One is that phosphorylation, autophosphorylation is occurring, and that is where, that is in the tyrosine residues in its cytosolic domain. And second thing is what? The activation of the intrinsic protein tyrosine kinase activity of the receptor, or you can say that ligands help to active the receptors. Clear? So first it helps to active the receptors, and second it helps in the autophosphorylation of these receptors. Clear? The activated receptor also can phosphorylate other protein substrates. So here autophosphorylation means they help the phosphorylation process in them, autophosphorylation. But sometimes the activated receptor, they helps in the phosphorylations of other substrates. And there are several types of proteins with which the activated receptors may interact. Okay, so when the receptors become active, they interact with different types of proteins or various types of proteins. And there are some proteins like that GTPase activating protein or GAP protein, phospholipase C gamma protein, or the SRC like non-receptor protein, tyrosine kinases. So what is actual thing? First, the ligand bind to the receptor. They first remain singly or as monomer. When ligand bind, they remain as dimer, and after binding of the ligand, what is happening? The receptor becomes active, and when receptor becomes active, they help in autophosphorylation. Not only autophosphorylation, they help in the phosphorylations of the other subproteins, or other, not proteins, other substrates. You can say other substrates. And so here the proteins are important which are interacted with the activated receptors, like these proteins. So here three types of pathways are seen for this receptor tyrosine kinases. Okay, one is the RAS-MAP kinase pathway. Second is the IP3 DAC pathway, and third is the PI3 kinase pathway. So three types of pathways are very important for these receptor tyrosine kinases. Or you can say that receptor tyrosine kinases or activated receptor tyrosine kinases, they functions their work, or they functions in three pathways. First of all, RAS-MAP kinase pathway, IP3 DAC pathway, or PI3 kinase pathway. So these pathways we have to learn in my here we just learn about the receptor tyrosine kinase pathways, kinases, or receptor tyrosine kinases means RTKs. What is the RTKs or its content, and what is the role of the RTKs. So in my next video I will try to explain about these three types of pathways in my